Assalamu alaikum. Hope all of you are fine. We'll discuss today the histology of the muscles. This is the part one, and in part two, we'll discuss the histology of the skeletal muscles. In this lecture, we'll discuss the classification of the muscles, what is the histological features of the smooth and the cardiac muscles, and a few of the medical applications. The word muscle is a Latin word which derives from the Latin word the musculus, which means the mouse-like. So it's a contractile tissue of the animal and is derived from the mesodermal layer of the embryonic germ cells. It contains contractile filaments that move past each other and change the size of the cell. The muscles, they are classified on the basis of the action into the voluntary and involuntary muscles. The skeletal muscles, they are the type of the voluntary muscles, whereas the smooth and the cardiac muscles, they are the involuntary type. And on the basis of their structure, they are classified as striated or non-striated. So the skeletal and the cardiac muscles, they have the striations, therefore they are called the striated muscles, whereas the smooth muscles, they are the non-striated muscles. Now the smooth muscles, they encircle the viscera. They are non-striated, involuntary, and they respond slowly to the stimuli. Their muscle fiber is elongated. They line the walls of the alimentary canal, the walls of the respiratory passages from the trachea to alveolar ducts, the walls of the arteries, veins, and large lymph vessels, the urinary and the genital ducts, the autonomic nervous system. They are under the control of the autonomic nervous systems. As they are involuntary muscles, and the hormones and the local metabolites, they can influence their. So you see the histological appearance of the smooth muscles. The smooth muscle fibers, they are elongated spindle-shaped cells with a single central nucleus. The nucleus is located centrally, and the sarcoplasm, which is the cytoplasm of the muscle, is called the sarcoplasm, is filled with the fibrils. The thick myosin and the thin actin filaments, they are scattered all around, scattered throughout the sarcoplasm, and are attached to the adherent densities on the cell membrane and focal densities within the cytoplasm. They appear smooth than the striated. So the actin and myosin filaments, they are present, but they are not arranged in the sarcomere units as in the skeletal muscles or the cardiac muscles. The smooth muscle fibers, they are bound together by irregularly branching fasciculi that vary in the arrangement from organ to organ. These fasciculi are the functional contractile units, and there is also a network of supporting collagenous tissue between the fibers and the fasciculi. Now you can see here the smooth muscles. This is the bundle of the or the fasciculi of the smooth muscles which are in the uncontracted state, and this is the bundle of the muscle fibers which are in the contracted state. And in between there is the connective tissue surrounding them. So in smooth muscles, the actin filaments, they are attached, the thin actin filaments, they are attached to the dense bodies, and on the plasma membrane you can see the within the dense bodies are they are functionally equivalent to the Z disc in the skeletal and the cardiac muscles. And on the plasma membrane you can observe the several invaginations, and these are called the cavuli, and these cavuli, they are the analogous to the T-tubule system in the skeletal muscle fibers. So the calcium is sequestered into these contractile elements through these T-tubules, and then is transported inside, and the action potential is generated. And the calcium binding protein in the smooth muscle is the calmodulin. So these are the photomicrographs of the smooth muscles. You can see the fasciculars or the bundles of the smooth muscle fibers. And this is a photomicrograph of the colon. You can see here the longitudinally cut smooth muscles, very beautifully seen the muscle fibers which are the fusiform and with the central elongated nucleus. And in between these muscle fibers, you can appreciate the endomysium connective tissue, which is in between them. And this is a layer between the, this is a longitudinally cut smooth muscles, and they are the transversely cut smooth muscles. And in between is the myenteric plexus, which is the autonomic supply to the smooth muscles. So as you know that they are involuntary, so they are supplied by the autonomic nervous system, and in between you can see here the myenteric plexus. So these are the transversely cut bundles or the fasciculus of the smooth muscles, and you can see there are the nuclei in some areas. You can see the nuclei where the some areas depending upon the section, the nuclei they are absent. So because if we cut a section transversely from here, so areas which have the nucleus, they will appear as such here, and the areas which come without the nucleus, they are as such. Now, the cardiac muscles, they form the myocardium of the heart. They are also involuntary. They are also striated. They are also under the autonomic control. Also, they are automatic and have the rhythmic contractions. So, their muscle fibers, they are single nucleated and branches and anastomosis with the neighboring fibers at intercalated discs. This is the cardiac muscles. You can see the striations in these cardiac muscles also, and these cardiac muscles, they are elongated uh, arrangement of the and they have a single nucleus, which is a single nucleus, but at the ends these muscle fibers, they branch and they are connected by the special the discs, which are called the intercalated discs. So the fibers of the cardiac muscle, they are arranged parallelly. They form branch at the ends to form the connections with the multiple adjacent cells, resulting in a complex three-dimensional network. The cardiac muscle fibers, they are long cylindrical cells with one or two nuclei. The nuclei are centrally situated like that of the smooth muscle. So this is another example of, you can see clearly the intercalated discs at the branching sides of the cardiac muscles, and this is showing you the uh, cardiac muscle fiber in which there is a nucleus located deep within the cell, centrally placed nucleus. Now, another photomicrographs of the cardiac muscles, you can see the bundles of the cardiac muscles in the heart. As you know, it is lies in the myocardium, and in between there are bundle of the Purkinje fibers here, which are the in conduct forms the conducting system of the heart. And you can see here these muscle fibers with the centrally placed nucleus, and these striations also, and in between there is the endomysium and the capillary endothelial cells of the capillaries also. Now, the cardiac muscles, sarcoplasm has the great amount of the mitochondria. You know that the cell membrane of the muscles, they are called the sarcolemma, and cytoplasm is called the sarcoplasm. And it is has a lot of the mitochondria for the energy demands of the cardiac muscle. The cells have an invagination of the T tubules. You can see here the invaginations of the T tubules and the sarcoplasmic reticulum. So this sarcoplasmic reticulum is analogous to the endoplasmic reticulum. So the collagenous tissue, they are found surrounding the individual cardiac muscle fibers. There is an abundance vascularization within this supporting tissue, which is required to meet the high metabolic demands of the cardiac muscle. So the cardiac muscles, they are joined by end to end by a specialized junctions, which are called the intercalated discs. Now you can see here these intercalated discs. These are the intercalated discs. You can see here The intercalated discs, they provide the anchorage for the myofibrils and allow the rapid spread of the contractile stimuli between the cells. Such rapid spread of the contraction, it allows the cardiac muscles to function, to contract as a syncytium. The intercalated discs, they are joined together by the special junctions, and there are three types of the junctions which can be identified in these intercalated discs. That is the fascia adherens, which are connected to the actin filaments to transmit the contraction, and then is the desmosomes, which connect the intermediate filaments of the cytoskeleton, and the gap junctions, and which are sites of the low electrical resistance that allow the spread of the excitation the rapidly and all the muscles, they contract as a syncytium. So this is showing you the cardiac muscles fibers, the myofibrils. The myofibrils of the cardiac muscles, they have also the light and the dark bands, the actin and the myosin filaments, and the sarcomere has the TZ discs and the T-tubule systems, which are at the junctions of the A and I bands. But the T-tubule system in the cardiac muscles is in the form of the dyad, not in the form of the triad. So in the medical applications, the cardiac tissue or the cardiac muscle tissue is damaged due to the lack of the oxygenation, due to the blockade of the coronary arteries and the myocardial infarctions and the angina, and there is a lack of the satellite cells in the cardiac muscle. So there is a little or no potential of the cardiac muscle cells to regenerate. Then the smooth muscles, they have benign tumors which are called the leiomyomas. And these benign tumors, when they are found in the wall of the uterus, that is the myometrium, which is made up of the smooth muscles, then it is called the fibroids. So it's all about the histological features of the muscle, that is the cardiac muscle and smooth muscles. So we'll continue with the histological features of the skeletal muscles. So if you have any queries, then you can contact me on the WhatsApp.